All right, so this is our last set of notes for the inheritance and genetics unit. So this will cover, I think it's like the last 3 pages of your notes. Starting is with what is biotechnology? So the word itself breaks down to bio meaning biology, the study of life, and then technology is the tech that's used for that. So biotechnology is going to be the use and alterations of organisms, cells, or biological molecules to make things. So that could be food, drugs, it could be like clothing, textiles, it could be altering plants to make better food sources. They could be used to make pesticides. So that is one use of it. It can also be selective breeding and genetic engineering. So these are going to be things where you can take bits and pieces of organisms and make new or better organisms with them. During biotechnology or using biotechnology, you can keep wanted characteristics and move those on to the next generation. And then characteristics or disease or traits that aren't necessarily wanted, those can be pulled out and then left behind. So it takes advantage of the naturally occurring genetic variation and passes those traits on. And then any traits that are unwanted, they can be cut out. The term hybridization is taking dissimilar or not similar organisms and bringing them together to make the best of both. So, an example of that would be like taking a buffalo and crossbreeding it with cattle. Normally, we can't do that, but in this case, we can. Another example would be like a liger. If you've ever seen Napoleon Dynamite, he talks about a liger, which is a lion and a tiger mixed. Naturally occurring, that cannot happen. You cannot breed a lion and a tiger, but using biotechnology and different methods, you can actually take their DNA and bring them together to make an organism. You can also do inbreeding, where you are going to take, and this doesn't mean like in the same sense of like cousins having babies together or siblings having babies together. This means that you take individuals with similar characteristics and you continue to breed them through to increase the likelihood of having those specific genes. So, like dog breeds, where you want them to have that like high level of the breed for that dog, that would be inbreeding. So, continuing to breed the same types of dogs over and over. So here's an example of a hybridization of a cow or cattle. This would be a bull and a buffalo. So notice how it has more of the head features of a buffalo or a bison, and then it has more of the body of the bull. And then here is an example of the inbreeding. And then on top of that, especially small dogs and small dogs with weird faces, so like a pug or a Pekingese or a Shih Tzu, that is the result of inbreeding over and over, where you're trying to maintain those characteristics within the breed line. And at some point in time, things got weird, but then those weird things were kept. So, biotechnology is also taking the process of making changes in the DNA code of a living organism. So, taking a specific organism's code and making those changes from an outside source. So, not actually crossing anything, but taking that one organism and making their DNA, either replicating it or making it completely different. Genetic engineering. So, genes, genetic engineering is like the creation of or like putting things together. Genetic engineering is something that is more direct and it's quicker. So, again, being able to take an organism's DNA, so this right here, genetic engineering, taking the DNA and actually just altering the DNA and not actually doing the process of breeding anything or putting these things together. What genetic engineering does for us, it helps us learn more about cells and genes. It also allows us to be able to figure out better treatments for diseases and disorders, being able to alter that DNA to make more specific medications. And we saw that in the gene doctors, if you watched that in class, where geneticists and biotechnologists and bioengineers or genetic engineers, they take a disorder that somebody has and they can make medications that are specific to that disorder. It also produces valuable biological materials and it can improve plants and animals for agriculture. So if you can alter like a corn DNA to make it so that the corn is not very susceptible to drought, or it's not susceptible to certain animal or like insects, it doesn't taste very good to like a grasshopper, then it makes it so that those plants are more likely to survive. So a part of biotechnology is going to be the extraction of DNA. So we will do this in class, but what you do during DNA extraction is you take the cells. So in our case, we're actually going to like chew our cheeks a little bit to get more cells. And then you lyse them, which means that you split them open using like a soap. Then you will add a an alcohol to it. And what that does is it'll cause the DNA to clump, and then you can pull the DNA out. Once you have the DNA out, then you can like isolate it, and then you can start really working with it. So during this time, uh, now if we're doing it in an actual lab setting, we get to use actual enzymes like protease, which is here. And then instead of using alcohol, ethanol is used because it's stronger. And then you can also centrifuge it, which means spin it really, really, really fast so that the DNA falls to the bottom. We can also use tools that cut, separate, and then read DNA. And using this information in this background, we've been able to basically code the entire human DNA or the human genome. And we'll get to that at the end of this presentation, talking about the Human Genome Project. But the Human Genome Project was the huge undertaking to look at human DNA. But you can also do this with dogs, cats, insects, bacteria. But when you do that, you are literally pulling apart the DNA base by base or nucleotide by nucleotide to say, okay, these are all of the base sequences, and this is what they code for. Restriction enzymes are going to be used to cut specific codes of DNA into fragments called restriction fragments. This is kind of a cool thing because when it does that, it creates this situation which is called a sticky end. And then those sticky ends can be put together with other organisms' sticky ends. And this is how you can put together different organisms. So you can take an example of this would be if you want something to become a bioluminescent. I know in AP biology, they do a bacteria where they take the bacteria, they splice it, which means they cut it. And then they add in the other sticky ends from a fluorescent bacteria or a fluorescent, jellyfish. So then the bacteria, when they grow on the culture plate, the bacteria will glow in the dark. So it's kind of cool. But they're also trying to do that with trees right now. They're trying to introduce DNA from a fluorescent animal or fluorescent algae. And they're trying to make it so that the tree will glow in the dark. Gel electrophoresis, we are actually going to do this in class. We are going to do some practice, and then we are actually going to separate the DNA strands of different organisms. So what this does is it splits the fragments of DNA, and then it runs them through a gel, which is made out of agar. And based off the size of the fragment, it'll allow for those fragments to like move down the gel, and it creates this banding. So it separates the DNA based on size. DNA, because it has that charge, that 5' 3' situation, and because of the phosphate groups, it carries that charge. So when you have the DNA pulled, so you pull the DNA, once you have that done, you will run it through a charged, like it's like a battery almost, and that allows for the DNA to run through the gel. The larger fragments they stay closer to the starting point. So in this example, these really bright bands, these are your starting points. So the bands of DNA that are bigger will stay closer. The ones that are smaller are able to travel faster. And so it allows you to see the similarities between different organisms and where those bands or those chunks are traveling. So this is how we make our electrophoresis work. So we put the DNA in a test tube containing the DNA polymerase. So remember, DNA polymerase's job is to copy the DNA. And then After that, we are going to, with that unknown template, or we use an unknown strand as a template, you can make these bands appear, and then you can use a chemical dye to make it very visible. And then using that information of these color-coded DNA fragments, you are able to determine, okay, this is where the similarities between our original or our known template versus our unknown template, this is where these similarities occur. And then you can determine how similar those two organisms are. Gel electrophoresis is also used in like paternity cases. So when somebody submits DNA to determine paternity, this is what they can do. So if they know, okay, here's child, they can run dad or potential dad to see how many of those bands match up. At least 50% of the bands should match. And if 50% match, because remember, you get 50% of your DNA from mom, 50% from dad. So if 50% match up, then that's how they determine paternity in a paternity suit. PCR, otherwise known as polymerase chain reaction, this is just a way to make a bunch of copies of DNA all at once, and then being able to take those copies of DNA and utilize them. So first thing that happens is DNA is heated to separate the two strands of DNA. Primers are added, and where those primers come in is like the part of the DNA that's going to be copied. So just very similar to how DNA replication has. So the primers are added, DNA polymerase comes in, copies that area between the two primers, and then that area serves as a template to continue the process. So this is how, using PCR, this is how you can make billions of copies of the same DNA over and over and over again very quickly, is because that same section, just that little section that's needed, is copied over and over and over again. Using using that same idea of the PCR, a person or a scientist could then take those sections, that specific section that we're looking at, cut it, and then again that produces the sticky ends. The sticky ends can then go to another complementary base sequence from another organism. DNA ligase binds them together, and when we do that, we call it recombinant DNA. So this is where you take, again, two DNA strands and you glue them together to make a new DNA strand. This can occur like in a lab, especially with major organisms, or this actually can happen in nature. So bacteria is an example of when recombinant DNA happens. Some more examples from nature: sexual reproduction. Again, you're taking mom and dads, you're putting them together, making new DNA as a result. Transformation in bacteria and viruses. Same thing, especially with viruses being able to transfer their DNA into the organism they're affecting. So this is where you can have two different organisms bringing their DNA together. This is also where you can make this genetically modified organism. So this means that this particular organism was created using separate DNA that is not coming together naturally or typically in a natural sense. A clone, this is going to be an organism that is made from an already there organism, and the DNA is going to be identical from a single cell. So there's three types of cloning. There's reproductive cloning, molecular cloning, and cellular cloning. So the reproductive cloning, this is when people think of cloning, this is typically what's thought of, where you make an egg from an already existing organism's DNA. Then you take that egg, it develops into an embryo, and then that embryo is implanted into the uterus, and then it'll grow an identical copy of itself. So this was the case with Dolly. Dolly was the first cloned organism that survived. And so in this case, the egg cell is taken from an adult sheep. Donor cell is taken from a sheep's udder. They combine them together, they fuse it, they make the embryo. The embryo is implanted into the mother. The mother has the cloned lamb. So this cloned lamb will be the exact same as the donor cell. So this lamb is the exact same as this sheep. Okay. Molecular cloning is when DNA is replicated using plasma. So you're just doing those chunks of DNA. And then cellular cloning is when you are cloning the cells themselves, typically in yeast and bacteria, those unicellular organisms. So here's just an example of that molecular cloning, taking just the DNA strands and then putting them back together. Hybridization versus genetically modified. Hybridization, you're not actually altering the set DNA at all. Hybridization is going to be like an example of if you have a tree that grows both oranges and apples. So you didn't actually change anything. You just introduced DNA to the cells that are then going to become these plants or these apple and oranges trees. So, and then using genetic variation, you can choose which options are passed down. But that is hybridization. So it's just like a hybrid. It's a mixture or a coming together of organisms. Here is as the years have gone on up to the year 2006-ish, 2007, the percentage of genetically engineered crops in the United States has increased significantly. And that's, again, it's because these crops are more likely to survive, they're more likely to last, and they can produce more outcome. So especially as the population of the world has gotten bigger, these percentages of organisms that are genetically engineered or these genetically engineered crops has had to increase to really stabilize the population of both the United States and then our surplus we send over to developing countries that otherwise may have people starving to death. Biotechnology can be used in health and medicine. So again, developing treatment options, developing medications, developing ways to make it so that if somebody has a genetic disorder, maybe making it so that their cells don't recognize that disorder anymore. Agriculture, genetically modified animals, genetically modified plants. And then biotech can also be used in human identification, identifying if somebody goes missing or they have amnesia and they don't recognize themselves, they don't know their name. Biotech can be used to identify that person. 23andMe and Ancestry.com also uses biotechnology to say, okay, this is where you come from. Okay, so with biotech and genetic engineering, again, kind of looking at the uses of this, health and medicine can be used to help prevent disease altogether. So making things like vaccines, antibiotics, different hormones that can cost for different diseases or different traits to make it so that they don't become an issue. Can be used in medical research, identifying new genes that can cause problems. And then also kind of working backwards to say where certain diseases or issues come from, where they're going, same with disorders. And then genetic engineering can also be used to help treat the disease so that, so an example of that would be like bacteria can be engineered to make insulin. And then that's how we can make like synthetic insulin or insulin that didn't come from a human, but it can be used to help treat diabetes. Gene therapy is also something that can be done where doctors and geneticists can work together to change a certain gene to make it so that it is now a treatment. So an example of that would be changing the hemoglobin gene and introducing it via a virus to make it so that the bone will create different bone marrow, so red blood cells. This is also the case. They are trying to work with this kind of example when it comes to Alzheimer's and being able to cure or heal Alzheimer's. That's kind of the premise behind it. In agriculture, genetic engineering can be used to make genetically modified food. And genetically modified food does have benefits. It's less expensive to make and it does yield more. And it could also make it so that the plants are less likely to succumb to disease, drought, insects, things like that. And it can also pack in more nutrition so that for example, an orange, if it used to have 10 milligrams of vitamin C, through genetically modifying them, maybe not has 20 grams of vitamin C. So it makes it so that you can get by with less is more. A specific example for gene modification in plants is that some plants will use, or excuse me, some geneticists have taken bacterial genes and it produces a protein called Bt toxin. For humans, Bt toxin is not an issue. It doesn't really mess with anything within our system. And for most animals are fine with it as well. But what this does, Bt toxin is toxic to insects, so it kills them. So plants that have this protein in them, if an insect tries to eat that plant, then it'll kill them. So through using genetically modified organisms and genetically modified plants, it has actually sparked an evolution inside of insects that tells those insects, don't try to eat this, it will kill you. So it creates that insect resistance for those plants, and it decreases the need for pesticides because it's like a natural pesticide. So some pros and cons for genetically modified foods. Pros is that they're safer, usually. They produce higher yields, and they don't need as much water. They don't need as many nutrients in the soil. They can grow faster. Some cons, however, is that there are some people that question the safety of these genetically modified foods. They also can have an impact on the ecosystem, and they can affect natural predator prey relationships among plants and animals. And then it can also force smaller farmers out of business. So the farmers that can't really afford to get started with these genetically modified organisms, they will eventually no longer be able to farm. The last little bit here is the Human Genome Project. The original goal of the Human Genome Project was to sequence the entire human genome. So to track out every single nucleotide, every single base, every single sugar, every single phosphate, and really just scan it out. In order to do, or in doing so, this would cause or allow for geneticists to identify new genes and see if there's a relationship between people having those genes and then developing diseases that are linked to them. It also allowed for scientists to compare everybody's genomes to each other to determine how much of the human DNA is similar to that of each other, and to identify the new genes that are unique to other organisms. So the Human Genome Project, this was something that was they started talking about it back in the 70s, but they really started putting it into play in the 90s. In 1993, there was a 99.9% accuracy of the genes. And it's kind of interesting with the Human Genome Project because in the early 90s, I mean, computers were a thing, but they did not have a method to really communicate with each other like we do with the internet. So the Human Genome Project was something that computers were helping with, but it was still a very slow process really up until the late 90s, early 2000s. Then it took off because now these labs that were all working together to put this giant undertaking together, the labs were able to communicate with each other quickly with the click of a button using the internet. So most of the Human Genome Project actually ended up taking place after the year 2000. But by 2003, the Human Genome Project that was completed was 99.9% accurate. And it accounted for most of our DNA. 2% of our DNA was consistent of 20 to 25,000 genes. So that was only 2%. And again, it took a really long time to get that 2%, but the remaining bit was done in the first few years of the early 2000s. I believe it was completed by 2000, 2004, 2005. So some issues with biotechnology is that, we look at things like traditional versus modern agriculture. What There are people that think what has happened in the past is beneficial for the future, and that's how it should be. And there's ethics. So again, talking about the ethics of changing agriculture, there's also ethics with changing our genome and having it mapped out. There is the question of, okay, if we can change a person's genes to make them less likely to have diabetes, at what point in time are people going to start changing their genetics or their embryos genetics so that they're taller or they're smarter or they have certain features, or we're making we could potentially be making a generation of humans that are specific for certain things, whether we want them to be athletes or we want them to be in the culinary arts or we want them to be really good readers. That's kind of where the ethics take into place. And the question is, what's going to happen to the human species once that starts happening? And that is something that there are labs in China that have started doing that, where the very wealthy in China can start to pay. But for these are the genes that we want our children to have. These are the specifications that we want. So there is at what point in time do they develop a board that says, listen, these are kind of the ethics behind it, and what are the consequences that are going to happen in the future with us tracking and changing our genome. So that is it for our biotechnology notes. If you have any questions, please let me know. Otherwise, I will see you in class where we can go over this a little bit more together. See you then.